Hi, my name is Katie Lee Huang, and this is the GI Physiology section. We're going to switch gears now and talk about the physiology of the GI tract. As you know, there are many hormones and secretory products that act on the GI tract and are produced by the GI tract. They all interact with each other. The information is organized into diagrams to help keep things clear. Gastrin is a very typical GI hormone. It is produced in the antrum of the stomach by the G cells, which you can see here, and it's released into the circulation. Gastrin works to increase gastric acid secretion. It increases growth of the gastric mucosa, and it increases gastric motility. How does it accomplish this? Well, gastrin works by acting on the enterochromaffin, or ECL cells, which then leads to the histamine release. This acts to increase parietal secretion of HCl. Gastrin also directly acts on parietal cells to increase acid release, but this is more minor compared to its effect on ECL cells. Well, how is gastrin regulated? Gastrin production is increased by stomach distension or alkalinization. Also, it's increased by the arrival of amino acids or peptides, in particular phenylalanine and tryptophan. This makes sense since if you have food arriving into the stomach, you're going to want to increase the stomach acid load. Also, vagal stimulation will increase G cell secretion of gastrin. However, when you have enough acid, you want gastrin production to decrease. This is signaled through the stomach pH, so it's a negative regulator of gastrin. Gastrin is also the hormone that is highly elevated in the Zollinger-Ellison syndrome, also known as gastrinoma. Cholecystokinin, or CCK, is the next hormone that we'll talk about, and it too helps with the digestion of food. It is produced in the duodenum and jejunum by eye cells. It increases both pancreatic secretion and gallbladder contraction, and it relaxes the usually contracted muscles of the sphincter of Oddi to allow these secretions to pass into the duodenum. CCK also reduces gastric emptying. Fatty acids and amino acids trigger the release of CCK, and it does act via neural muscarinic pathways to cause pancreatic secretion. Secretin is produced in the duodenum by S cells. What is its function? Well, secretin's functions are to reduce the amount of hydrogen ions in the small intestine, so pretty much act as a negative regulator of the acid. Similar to CCK, it works to increase secretions. Specifically, it increases pancreatic bicarb secretion as well as bile secretion. This is important because the bicarbonate increases the pH in the duodenum so that the pancreatic enzymes can function. In addition to this local effect of increasing the pH, secretin also heads to the source in the stomach and reduces gastric acid secretion. Secretin is triggered by low pH and fatty acids. Next, let's talk about glucose-dependent insulinotropic peptide, also known as GIP, or gastric inhibitory peptide. GIP is secreted both from K cells in the duodenum and jejunum. This hormone has both exocrine and endocrine functions. It decreases acid secretion, and it also increases insulin secretion, hence its two names. So what stimulates GIP release? Well, GIP is the only GI hormone that is released in response to fat, protein, and carbohydrates. Importantly, an oral glucose load is actually used more rapidly by the body than an equivalent IV load. Why is this? It's because of GIP's effect. Next, somatostatin is a hormone produced by D cells in pancreatic islets and in the GI mucosa. We already showed you where the D cells are within the islet during the endocrinology lectures. Can you remember where they are in relation to the insulin-secreting beta cells? Well, the D or delta cells are interspersed throughout the islets, whereas the beta cells are located centrally, which you can see here. You may recall that the function of somatostatin is basically to reduce the release of everything. This hormone decreases gastric acid secretion and pepsinogen secretion. It also decreases pancreatic and small intestine fluid secretion. It slows contraction of the gallbladder to reduce bile secretion, and it also acts to reduce insulin and glucagon release. This is an inhibitory and anti-growth hormone. Somatostatin release is increased by acid content, but it's decreased after vagal stimulation. Vasoactive intestinal polypeptide, or VIP, is released from the parasympathetic ganglia and sphincters, as well as the gallbladder and the small intestine. VIP is responsible for increasing intestinal water and electrolyte secretion and relaxing the intestinal smooth muscle and sphincters. This function of increasing fluid secretion can be a source of pathology in the VIPoma, which presents with copious diarrhea. VIP in the normal state is increased by distension or vagal stimulation, and it's decreased by adrenergic input. Okay, we just have a few more to go. Nitric oxide is responsible for smooth muscle relaxation, including at the lower esophageal sphincter. You're probably more familiar with its role in cardiovasculature. The clinical correlation is that without NO secretion, you may develop achalasia. Finally, Motilin is the last hormone we'll mention, and it comes from the small intestine. Motilin is the hormone that causes the migrating motor complexes, which occur more frequently in the fasting state. Indeed, Motilin receptor agonists can be used to stimulate peristalsis. Let's do a flash quiz. Does somatostatin lead to increased or decreased pepsinogen secretion? And does it lead to increased or decreased gastric acid secretion? Well, it's decreased for both. Somatostatin, remember, is an inhibitory hormone. Okay, let's now go on to some GI secretory products. The GI tract also has a number of important secretory products, namely acid and bicarbonate, as well as intrinsic factor and pepsin. Intrinsic factor is produced by parietal cells, which you can see here, in the stomach in order to allow for uptake of vitamin B12 in the terminal ileum. Autoimmune destruction of these parietal cells can cause chronic gastritis and will prevent production of intrinsic factor, thereby preventing absorption of B12 in the terminal ileum. If you think back to hematology, what disease is caused by a lack of B12? That's right, that would be pernicious anemia. Gastric acid is produced by these same parietal cells, and its secretion can be increased by histamine, acetylcholine, and gastrin, and it's decreased by somatostatin, GIP, prostaglandin, and secretin. We already mentioned most of those in the last section. Gastrinoma, as you recall, is also known as what? That's right, Zollinger-Ellison syndrome. Zollinger-Ellison syndrome is noticed as ulcers when the very high acid content harms the stomach lining. Pepsin is another product of the stomach. It's produced by the chief cells, which you can also see here. It is first secreted in the inactive pepsinogen form, which is then cleaved into pepsin. Pepsin is required for protein digestion. Pepsinogen secretion is increased by vagal stimulation or local acid. Lastly, bicarbonate. Bicarb is produced in many different places, especially in the mucosal cells, and of course its purpose is to neutralize acid. This neutralizes the acid of the stomach so pH-sensitive enzymes can be activated. Bicarb secretion is stimulated by pancreatic secretion as well as biliary secretions. Okay, let's do another flash quiz to make sure we understand what we've just gone over. What substances are secreted directly into the lumen of the stomach? That's right. As you recall, it's mucus, pepsinogen, intrinsic factor, as well as HCl are all secreted into the lumen of the stomach. Let's summarize what we've learned so far and put it all together. It might be helpful to go back over some of the things we've just talked about, but this is an excellent picture that provides a wonderful summary. Here you can see the parietal cells, chief cells, and G cells in the stomach. What does the parasympathetic signal coming down the vagus nerve activate? Yes, it activates parietal cells as well as G cells. The parietal cells release acid in response to three stimuli. First, vagal acetylcholine release. Second, gastrin via the G cells that reaches the parietal cells via the circulation. And also from histamine that is released from ECL cells in response to gastrin stimulation. This indirect gastrin effect through histamine is actually more powerful than gastrin's direct effects on parietal cells. The parietal cells are also releasing intrinsic factor. The chief cells, on the other hand, release pepsinogen. How is pepsinogen activated? Again, recall that pepsinogen needs the acidic environment to be converted to pepsin. A side note on the schematic is that whereas the vagus releases acetylcholine into the body of the stomach to activate parietal cells, the molecule that activates G cells in the antrum is actually GRP, or gastrin-releasing peptide. You can also see here on the left side the other cell types of interest. I cells releasing CCK, S cells releasing secretin, K cells releasing GIP, and in the pancreatic islets, D cells releasing somatostatin. Now we have an up-close diagram of the parietal cell. It is important to know the mechanisms of acid release so that you can understand the types of drugs that can interfere with acid production. There are 3 ligand receptor pairs that increase acid production and 3 that decrease production. The 3 that increase acid are acetylcholine, gastrin, and histamine. And the ones that block acid are prostaglandin, misoprostol, and somatostatin. As we just mentioned, the vagus can release acetylcholine, which activates M3 receptors. And it also releases GRP to activate G cells into releasing gastrin. Atropine can block the effects of acetylcholine, but not gastrin. What does this mean? Well, it means that gastrin can still directly stimulate the CCKB receptor and indirectly stimulate release of histamine to act on H2 receptors. We also mentioned that histamine is more important than gastrin, so the histamine antagonists are quite useful drugs. Cimetidine is one useful example. We'll talk more about histamine antagonists during the pharmacology portion of the lecture. Now, if you look inside the cell at the mechanisms by which these receptors signal, you see that acetylcholine and gastrin activate G-protein-coupled receptors that use the IP3 pathway to upregulate the hydrogen-potassium ATPase, whereas histamine increases cyclic AMP to upregulate the hydrogen-potassium ATPase. The molecules we mentioned that decrease acid production, such as prostaglandin, misoprostol, and somatostatin, all signal through an inhibitory G protein that reduces cyclic AMP production. Before we move on, note the most important class of acid medications, the proton pump inhibitors. This diagram really points out why drugs like omeprazole are so useful. While histamine antagonists can block 1 out of 3 inputs to the parietal cells, there are still other pathways to stimulate acid production. But if you block the acid pump directly with a PPI, then it doesn't really matter what stimulation the cell is or is not receiving. Let's do a quick review flash quiz. How does acetylcholine stimulate acid production by parietal cells? Remember, it activates an M3 receptor on the cells that directly stimulates acid secretion via a G protein cascade.